Hello and welcome everyone. Today's session of the Science Policy Forum for Biodiversity from the International Union of Biological Sciences. I'm Simon Häusler from VBio, and I will quickly introduce you to Zoom and the functions we're going to use. So during the talks, you can already send your questions into the Q&A. We will prioritize the questions by their likes. So whenever you find a question that is already asked that you're interested in, please also just like that question instead of typing it into the Q&A again. Then you can use the chat for thanks or for other things, that also feedback or recommendations. And then I will hand over to Carl-Josef now. While Carl-Josef is introducing the IUBS, I will do a live poll to see where you're coming from and what your professional occupation is. Thank you.
Okay, thank you, Simon, for organizing and managing this webinar. So I'm Carl-Josef Dietz, the president of the IUBS, and we are very happy that first you join. And second, that we have now 3 webinars in consecutive order. Yesterday, we had one about invasive species, so you could look at it in the web. And today we have one which is very important as well, digital sequence information and biodiversity research. So I would say digital sequences are the currency of modern biology. And though digital sequences are very much decisive for our future success in dealing with many challenges, and so I think we really have to find the right regulations to deal with digital sequence information and how to share benefit. And so now I would like to move. Just a short introduction. I will make it as fast as possible. Because our target time for finishing is something like 4:50, so in 1 hour and 50 minutes, because you're on different time zones, we hope to finalize our discussion. So IUBS was established in 1919, so we are 100 years old this year. And we have 110 national scientific and institutional members, for instance, national academies and international scientific associations and societies. And just to give you an idea what our aim is, our action plan, we have major areas of work. And these are, for instance, scientific programs that receive seed funding. It's not millions of euros, but seed funding. And so we are involved in biodiversity and health big data, and you see already big data and today's topic, digital sequence information, that coincides. And this will be very important in the future, as I mentioned. So we are organizing now, I will show on the following slides, the Science Policy Forum for quite a while, together with other collaborating institutions. We have a program running on pastoralism, what is a very important, economically very important activity. Many people on this globe rely on pastoralism and the income they generate from that. Zoonotic diseases, in fact, not just Corona and COVID-19, but before, that is very important topic. And I would point to unified language, because that is very, very, very significant. So comparing it with the International Union for Pure and Applied Chemistry, where they have the CAS nomenclature of compounds, we have to strive for a common language in taxonomy, for instance. And we are engaged in the International Year of Basic Science for Sustainable Development, which is over. But now we have the International Decade that has started, and we are very much engaged in getting this International Decade running and cooperating with the other stakeholders. We are also involved in education, for instance, climate education and ecological education. I would also like to mention equal opportunities and gender equality. And so you can imagine that cannot be done alone. So we collaborate, and I would call IUBS as a voice of global unifying biology. And so we are interacting with UNESCO and UNESCO-related organizations like the Convention of Biodiversity and the International Science Council. And I mention here just as an example, we could mention many more, INQUA, which is an international union for quaternary science, because this is an example where we now try to support early career scientists to come together. So this is also a very important activity to support young researchers in their career. So now we are already closer why we are organizing today's webinar. It is in context of the Science Policy Forum to take place in Cali, Colombia later on in October. And naturally that is CBD. And you see that the first to fifth science forum were run starting from 2006, and the last one in 22, and now the sixth ones, and we will also organize it together with collaborating institutions. And in this context, we thought we should start with this webinar. As I mentioned yesterday, invasive alien species, today digital sequence information, and tomorrow there is going to be a webinar on indigenous peoples in conservation of biodiversity. That one will not take place as far as I understand. Okay, so today we have 4 people presenting thoughts, ideas, concepts, and 2 moderators. And in fact, 1 moderator is also presenter. So as I mentioned, I'm Karl-Josef Dietz from Germany, and I'm a biochemist and physiologist of plants, and I will moderate the first part. And that is where I would stop with my presentation. And I would like to introduce the first scientist who is going to give a talk. It's Douglas Miano, and he is professor of plant pathology at the Faculty of Agriculture, University of Nairobi. So we are very happy that he joined from Kenya. He has more than 20 years of experience in training and research, focusing mainly on plant virology. He is a Kenyan principal investigator and lead scientist in the development of transgenic cassava. I also saw a very nice publication from you, Douglas, on this topic with resistance to viruses. So he is member of the African group of negotiators for the Convention on Biological Diversity, CBD, and has presented Kenya and the African group in CBD negotiations, for instance, on risk assessment, synthetic biology, and digital sequence information, the topic of today. And if you look into his publications, I should make it short now, he publishes on risk assessment, genetic diversity of cassava, land races, I already mentioned it, or other virus diseases that cause a lot of losses in agriculture. And so we have to better understand them and to fight against them. So Douglas, it's up to you. Thank you very much for joining.
Thank you, Professor Dietz, for the invitation and the introduction. I hope I am clear.
Yes, it's good.
All right, thank you. So I want to share my slide and let me know when it can be seen.
Yeah, now we see your desktop. Now we see your presentation in the pre-mode, and now it's perfect. Yes.
All right, thank you. As I already said, my name is Douglas Miano. I work for the University of Nairobi, where I teach plant virology, both to undergraduate and graduate students. And today I want to talk a little bit on what we have been working on, especially the research in plant pathology and specifically in plant viruses, and how digital sequence information has been a key part in terms of this research that I have been doing. So the slide's moving.
Let's wait. Not yet.
They seem not to move.
No. Could it be that the Zoom window is in front of the presentation?
Yes. How do I take it back?
Just double click into the current window, and then you should be able to see a taskbar. And then, yeah, here we
Perfect.
All right. Thank you. Thank you.
Welcome.
So, as I have said, I'll be talking about the use of DSI in plant microbe research, and specifically, I'll be focusing on the work that we've been doing on viruses and why DSI is important, and especially how we are able to access it. So I'm going to look at the different areas of where we have been able to do the research on, and I'll basically look at what we've done on diversity studies, not biodiversity, but diversity, especially of the viruses, and how the viruses, some of the studies we have done on the interactions of the pathogens with the plants, some of the work that we've done on discovery and the evolutionary research, some of the work that we've done more so in crop improvement, and especially for disease and pest resistance, and nutritional enhancement. So as has already been said, a lot of examples, or the examples I'll be giving will mainly focus on cassava, which is the crop that I've been working with for some while now. And I've just given some photographs down there to show how the cassava looks like for those who may not have seen the cassava. You can see at the far left, the crop when it is looking healthy. And at the far right, you can see the crop that is infected when it is infected either with the viruses in the roots or the viruses in the above ground biomass, the type of symptoms that are expressed. So let me see it. Okay, yep. Beautiful. So looking at the type of diversity studies that we have done, and mainly I was giving the example of the viruses that we've been working on, especially in infecting cassava. And more so, there has been one of the viruses that has been, it has been endemic in the region, East African region, for many years, but as a minor disease. But slowly it went into the highlands where the disease is, where the cassava production is more. And when it went into the highlands, it all of a sudden became a major pathogen. It was actually classified as one of the 7 most dangerous diseases in crops in the 2000s. And because of this, we had done little studies on the diversity of the virus, so we knew very little about it. And so there was no much sequences or digital information that was available for the virus. And for that purpose, it was very difficult for us to understand the diversity and also look for ways to manage the virus. So for that purpose, around 2009 and 2010, we had to embark on work to have the full genome sequences for the virus. And we had to go and do the surveys for the viruses where they're infecting the cassava. You can see a cassava that is infected with the virus and how the roots were damaged. And eventually we had to do the sequences. And after those sequences, we are able to do the diversity studies. And these diversity studies could help us to know the variability of the pathogen. And with this variability, then we were able to come up with strategies of its management, which I'll talk a little bit more as I mentioned about the strategy that has been working for now. But the critical thing here is to indicate that the digital information was very, very critical for us to be able to understand the diversity and therefore come up with a strategy for the management of the virus. So directly meaning that when we are talking about digital information, as much as we are talking about conservation, it also has a key contribution towards food security. So the next part of the work that you have done, and this we had started doing it early on in my career as a scientist, was interaction studies. And I did my PhD looking at the viruses that infect sweet potatoes. And there was one critical disease that is known as sweet potato virus disease, which is a combination of two viruses, the sweet potato feathery motor virus and sweet potato chlorotic stunt virus. And these two viruses result to what we call SPVD, or sweet potato virus disease, which is a complex of the two, a synergistic interaction of the two. And the complexity here was looking for genotypes which were resistant to the disease, because we realized that when one virus comes in and infects the sweet potato, that would break down the resistance mechanism for the next virus. And as long as the two viruses were there, it was very difficult to have plants which were resistant. And you can see down there, we have a plant that is showing sweet potato virus disease, and one that is showing an infection of only one plant. So we did already the sequences for the viruses were available for the two viruses. And what we wanted to know is how which genes were being affected, or the genes which are involved in the interaction of the host plant with the combination of the viruses. So these studies were done back in 2009, again showing that part of the studies were done were very early, and we are able to show the gene expression studies, or which genes were responsible for the expression of the disease. And this was also important in terms of when we are trying to develop genetically modified crops for the resistance to the disease. This work has not been yet completed. It has been a complex studies, but basically showing that digital sequence information are also important when you're dealing with interaction studies. And this is important, especially as you identify the genes which could be of interest. And especially now that we are having technologies that have come up, for example, genome editing, we know which genes that you can be able to target if you want to change the expression of the diseases. So the third type of study that we do, this includes the pathogen discovery and evolutionary studies. Again, a lot of viruses and other pathogens affect crops, especially in the tropics. And we do have a wide diversity of viruses, and some of them we don't even know them. We don't know their effect on the plant. So we might be trying to control one virus, and yet there could be other viruses that are also helping in a synergistic interaction or in breakdown of resistance, and being able to identify and know which viruses are actually affecting the crop is of critical importance. So some of the studies that you have done, for example, I had recently the student graduated last year. He was from Zambia, and he was doing the discovery of viruses that infect common beans, the Phaceolus vulgaris in Zambia. And for this, we are able to identify quite a wide range of viruses that actually were infecting about 6 or 7 of them, which were found to be new, infecting beans in Zambia. So again, this is important for disease management, and eventually because you know which viruses you are targeting. It is interesting because sometimes you think that the virus or the pathogen that could be most prominent in the region, and you end up realizing that the pathogen of interest, or the one that you focus on, is not what you've been thinking about. And this was the case, for example, in Zambia. We realized that the virus that everyone was working on was not the major pathogen of interest. And it was another virus came up to be the one that should be targeted more, even in terms of development of resistant materials. So the work that I have spent a lot of time on is on the development of resistant plants, and especially using genetic engineering. And we have used the studies, the genetic information, for example, in marker acid cell selection. Some of my colleagues have been working specifically on that. There's work that is going on genome editing, but my focus has mainly been on the work of genetic engineering. My slides are still can be seen?
Yes.
Okay, thank you. So I was explaining about the genetic engineering work that we have been working on, and heavily also depending on the sequence information of the pathogens. This is for the picture that I had shown earlier of the cassava that is infected with one of the viruses that affects the root, the cassava brown streak disease, which is caused by two viruses, the cassava brown streak virus and another species which is known as Ugandan cassava brown streak virus. And as I hinted before, this is a virus that has been in the region for many, many years, for close to 100 years. But until the year 2000, it's when the main studies started because it caused an epidemic in the main cassava production regions. When the sequencing were done, initially we only had partial sequences for the UCBSV. And that was the only partial sequences were there, mainly the coat protein. And when the outbreak came, we wanted to understand what is it that is resulting to this, especially because we could see the symptoms where the expressions could be different, indicating some levels of differences. And when the sequences were finally done, we found that there were actually two viruses. So the UCBSV became actually the most predominant species, especially in the hybrids. So, and we had to look for a way of being able to manage this disease. Unfortunately, there were no resistance in cassava relatives. And since also it was a disease for which was restricted to the East African region. And so when we tried to look for sources of resistance, we couldn't be able to get it, but we decided to use for genetic engineering. We tried different techniques and eventually we settled for gene silencing. We identified a common lesion that was highly conserved, the coat protein, as you can see there, and we developed a construct, which is eventually is an inverted repeat. And this gives a hairpin. So eventually when it is put into the plant, the construct is put in the plant, it forms a hairpin, which triggers the silencing mechanism. And when we used that, we were able to come from what you can see from the extreme left, cassava roots, which are like extremely damaged. And we've been able to come up with very clean cassava roots, which are very, very healthy and as expected. So this has been type of work that we've been doing, looking at genetic interaction studies. A lot of other pathogens we have already done with similar work and with other colleagues. But basically for today, I wanted to highlight on what we've been doing and showing the importance of the digital sequences to the work that we are doing even in virology. So as we talk about maybe the importance of access and the products that are coming out of the market, we know that the basket is wide and it may not necessarily be on the products that could be at the high level markets, but there are also products that are within the farmers. So some of these products are not necessarily going to be sold in the big supermarkets, but it has a direct bearing on the farmers at the local level. So the other part that we have been also been working on using digital sequence information is on disease diagnostics, either using coming up with PCRs or primers that can be able to help us to detect those viruses, whichever type of molecular-based diagnostic techniques, whether they are RAMP or many others, or the others that could be available. Coming up with serological techniques has also been helpful. This is one of the techniques that you would want to have, especially for screening of seeds to confirm that the seeds are free from diseases. We have also been coming up with biosensors. We have one of my projects is to look at developing biosensors for detection of viruses. The other one is, of course, diagnostics by sequencing. So all of these different diagnostic techniques require us to have sequence information for the different pathogens. So, and at the end of the day, even when we are dealing with the farmer who is in the field, he or she would want to have materials which are clean from diseases. And for that, we need to have techniques that are easy to use, that are not too expensive, that are not going to use a lot of equipment and expensive equipment or use of electricity or power, especially in the remote areas. So all that we try to come up with easy techniques and again, we fall back on the use of digital sequences. So to summarize my presentation is just to highlight the final notes of why it is important for us to have this digital information. And we are saying is that the sequences are important, especially when you're dealing with the interactions and research and coming up with products and diagnostic methods. So, and this has a direct bearing also on food security. So, and this means that the decisions that we make, we also realize that it's not only about money that may be coming to the people's pockets, but it also has a bearing directly with the farmers who are growing crops for subsistence. Because at the end of the day, regardless of the crop that you are dealing with, you need to manage these diseases, and you have to have a way of being able to tell that you are dealing with high quality seeds or high quality planting materials, and therefore, and even when they are high quality, they must also have resistance, and so that then you can be able to get the yields that you needed. I want to end my presentation there and say thank you.
Mm-hmm. Thank you so much, Douglas. I don't know whether any question arrived in the chat. Maybe I can first ask, cassava, I'm quite sure, does not belong to Nagoya Treaty, but to the International Treaty on Plant Genetic Resources. So it's excluded from Nagoya, right?
Yes.
So cassava is one of these crop plants that is excluded. And what about the viruses and the pathogens? It's also excluded from Nagoya, I assume.
Not directly, because these are pathogens which could be found anywhere. Some of them are actually coming from the wild relatives, which could be in the forests. So they may not necessarily be direct, but as long as you're talking about a genetic resource, then you are also thinking about the pathogen from whichever source.
Mm-hmm. Okay. Are there any other questions? So our idea was to have now only a very few questions, 1, 2, maybe maximum 3, and then to have a summarizing discussion at the very end. So is there any question? Do you see it, Simon? Is there a question?
So far, there's no questions. Maybe people are still typing. In the meantime, thank you, Douglas, for your talk. And since you already asked before the session, I can quickly tell you that there's a lot of different people in the people listening to us and watching this webinar. It's research managers, researchers from academia. There's educators, bachelor and master students, PhD students, a postdoc fellow, and 25 people clicked on other. Maybe you can use the chat box and tell us what you do if you're not listed in the things that I was reading aloud. And people are coming from Africa, Asia, Australia, Eastern Europe, North and Central America, South America, and the Caribbean, and Western Europe, most of them actually. So far, I still cannot see any questions. Maybe.
Okay, so maybe, Douglas, we go then later on to the general discussion. Yeah. And how, for instance, Kenya looks at DSI in Nagoya, maybe. I don't know whether we have time to talk about it. So right now, I would thank you for your fantastic presentation. This is very important work. And now I have to find bio of Jens.
There's actually one question now. Sorry if I... Is an infected cassava product dangerous for our health?
Yes, and a good question. No, it is not dangerous. Most of the viruses that infect plants, almost all of them actually are not do not infect human beings and animals. Mm-hmm. So they are not necessarily causing infection to humans, but they reduce the yields. So that means that for people who depend on cassava, and especially for some of like this cassava brown streak virus, it is expressed in the final product so that you lose it at the end of the season.
Mm-hmm. Okay.
Thank you.
And there's actually one more coming. Why is comparative analysis important?
Yes, it is important because some of them is, for example, when you're dealing with the value, some of the information that you generate you may not necessarily know the value until you compare it with other studies that have been done. So it's basically to confirm what other people have done and know what it is about. And as much as possible, when you're dealing with only one sequence or two sequences or a few isolates from one region, it may not give you conclusive information. So you need information from other places, which can help you to be able to come with conclusive information on what it is that you're dealing with, whether it's the pathogen itself or whether it's the genes within the plant. And that can help you even, for example, in confirming the diversity.
Okay, great. Thank you so much. So we now move on to Jens Sandström, and he is Associate Professor of Plant Physiology as well at the Swedish University for Agricultural Sciences, SLU in Uppsala. He is also a co-chair of the Agricultural Technology Working Group of the European Plant Science Organization, EPSO. EPSO also tries to do lobbying at the political level in the European community. He has coordinated the implementation of a workflow for handling access and benefit sharing under the Nagoya Protocol at his home university. And we will later on hear Sebastian, who is also involved in this kind of setting it into practice. He will share his experience from a researcher's point of view, and he aims for raising awareness about access and benefit sharing adhering to the Nagoya Protocol. And what's coming along now with DSI, that will be very important. So he works on very important topics like low methane rise or gene annotation in Norway spruce, and blood and flower development, I found out, so a broad spectrum of research. Jens, it's your floor.
Thank you very much. So let me just share my screen. I hope you can see my presentation.
Perfect.
Thank you very much for that nice introduction. I also want to thank Douglas for giving a nice overview of the importance of DSI. So I have, for the past 10 years, been involved in different aspects of sort of both basic science, but also giving scientific advice to policy. And sort of lately, the handling of genetic resources and DSI has come up as a issue or as a point of interest from my turn. So I would like to share you some of my thoughts on this and maybe set the scenes for Sebastian and Amber later. So this will not be a talk about low methane rise, although I can talk about that also, but I will talk more about policy things here. And I will focus on plants because I'm a plant physiology. Of course, much of this is also of relevance for microbes and other organisms. But so my talk will mainly be a plant-based talk. So when we talk about plant genetic resources or resources from plants, I mean, you can sort of view of it sort of as the sum of the hereditary material. So all genes that are present in the plant, and of course one thing that has come up as a striking when we know more about plants and also do a lot of more sequencing is that quite a lot of genes are common to among plants and of course among other organism types also. So the view of I think DSI is sort of changing how we look at species, because on the species level or on the gene level, maybe not the species concept is not really of relevance anymore. Anyway, so if you look at the genetic resources, there is a lot of different regulatory frameworks that regulate how we handle this as plant biologists. So we have in the bottom end corner have the CBD and the Nagoya Protocol, which is regulated by where we sign PIC and MATS. I'll come back to that later. But we also, as alluded to in the previous question, we also have something called the International Treaty for Plant Genetic Resources for Food and Agriculture, or short, the treaty, because it's so long, which regulates access and benefit sharing from a subsection of the crop species that we use. But then, of course, there are other regulatory frameworks that regulates breeders' rights, that's UPOV, or in certain cases also patents like TRIPS. So I will focus then this one on DSI, but one important, on the two first ones, the treaty and the CBD. But one important point when we talk about DSI is that the issue about how to view DSI and how to sort of maybe regulate it or not regulate it are ongoing in all these regulatory frameworks. So it has the DSI question has bearing on all four frameworks, and probably more regulatory frameworks if you look outside of the plant field. So DSI is central, and in this question, when we look at this, and it's also sort of an emerging issue that we need to handle. But in order to sort of understand why this may be a problem, I would like you to sort of take you through how we handle access and benefit sharing within the Nagoya Protocol when it comes to physical samples, and also with the treaty, to see that already that process is fairly complex and it's not all that easy to adhere to actually as a scientist. So we start with the Nagoya Protocol, and in the Nagoya Protocol, which is sort of a part of the Convention for Biological Diversity, there is a paragraph about the fair and equitable sharing and of the benefit arising from the utilization of genetic resources, including appropriate access to genetic resources. Connected to this paragraph, there is an Article 15, which in a way is a paradigm shift, because I think previously we sort of looked at the genetic resources as a global resource, but in this Article 15, they state that genetic resources are no longer common heritage, even though genes might be similar in different species. The genetic resources are no longer common heritage, and states have the sovereign rights over the genetic resources and may regulate access and utilization. And that, of course, then puts some administrative burden on us as biologists if we want to use these resources. So if you are a scientist and want to access genetic resources, you have to check with the provider country, and there is a special website where you can check which type of rules is for different countries, and then you have to seek permission and to access these. It's called prior informed consent, and then you have to negotiate terms, mutually agreed terms on how to use these resources. And then you have to, once you have done that and you have done your research, you have to declare that you have fulfilled all these requirements. And this might seem quite straightforward, but it could be actually quite, well, it's not overwhelming, but it's a bit of an administrative task. So a simple example from a project that we are running at my home university is where a researcher studies evolution of gymnosperms, so conifers and different, yeah, so not only conifers but other gymnosperms also. And she collects has collected material from the Philippines, from Mexico, and from Nepal. And so in all these cases, what you have to do then is that you have to contact that country, have to find out what kind of different rules are in if they are part of the Nagoya Protocol, and if they have national legislation. So when this project started, Nepal had not yet signed the Nagoya Protocol. Singapore had signed it. Mexico had not signed it and had national legislation. So there was sort of there was some sort of paperwork that has to be done that was specific for each country, all on national all on national sort of languages. So it was not sort of something that you could just send a master student to do collection. You have to actually do some real paperwork in this case, and that of course is a change, I think I would say. Okay, so to aid in this flow, we created a routine at our home university, which basically follows this rather simple scheme where you as a scientist has to prepare. You have to do sort of the basic groundwork yourself because you know the research. So you have to, which countries am I collecting from? Do they have any access and benefit sharing legislation and so on? And then you have to contact that or contact the country in place, and then you have to then you can get help from Sebastian, actually, who is going to talk later about drafting contracts, and then you get help from the central administration to register and to do the declaration. So this is also a scheme that we try to follow and try to sort of inform our scientists of. In practice, it looks a bit more complicated, actually, because there is a fairly complicated scheme that you have to follow, because sometimes certain countries, they have signed the protocol, but they have not actually put any laws into place. There might be a focal point or not, and there might be overlapping national legislations within the Nagoya Protocol. So we have then developed this more elaborate scheme where you can actually go and follow and how to navigate in this one, and this has to then to be done for each sample and each thing that you want to bring into the lab. Okay, so if we go, so this is the Nagoya Protocol, and what we do here is that we negotiate bilateral agreements with individual countries. If we go to the international treaty, then it's not bilateral. So the international treaty then covers crop plants, not all crop plants, but some crop plants. But the international treaty is multilateral, meaning that you don't have to sign individual contracts with countries, but there are standardized agreements, and there is what this system is financed by companies who pay into a common fund to facilitate this sharing of genetic resources. So it's not the scientists themselves, really, as basic scientists, that do the payment in this one. Okay, so Nagoya basically regulates all genetic resources and traditional knowledge, whereas the treaty regulates certain food crops. These are bilateral agreements in the Nagoya, whereas it's multilateral. The agreements in the treaty, the Nagoya, the documents are called PIC and MAT, and it's SMTAs in the treaty. and benefits should be shared with the provider country in case of the Nagoya, whereas it's a common fund in the treaty. So those are the difference. Carl Josef, how am I in time? I don't hear you, so it's
yeah, sorry, fine. I had to activate my loudspeaker. I think 2-3 minutes, maybe. Is that fine?
Okay. Yeah. So basically, if you as a scientist want to access material, you have to sort of make sure that or find out if the country that you want to bring something in has signed and ratified the Nagoya Protocol, and also to see if there's any national systems on top of this. And then, of course, you have to find out if your species is listed in the international treaty or not. And then, so that might then be different from different countries and different species. So, as an example, tomato from Bolivia, then it's the Nagoya Protocol, because Bolivia has signed the Nagoya Protocol, but tomato is not on the list, is not on the treaty. Rice from China is also the Nagoya Protocol, because China has not signed the treaty. So therefore, if you take rice from China, it goes by the Nagoya Protocol. Other countries, it goes by the treaty. Plant material from Brazil has is covered by national systems. Plants from Denmark, if you work, then they have signed the Nagoya Protocol, but they don't have any access legislation, meaning that you can go there and collect as you like, or maybe sign MTAs. And that's the same for a lot of European countries have it that way. We have signed the Nagoya Protocol, but we have not implemented any legislation for this. I will skip the other ones, and I will jump quickly to the digital sequence information now, because now I've sort of tried to painted this picture of a rather elaborate system where you actually have to know what you're doing. And then, of course, if we talk about DSI, then everything becomes even more complex. So what happened in the last COP meeting, and I'm sure Amber will come back more on this, is that there was a decision that taken that there should be a mechanism developed for access and benefit sharing from the use of DSI, and that this mechanism should be a multilateral mechanism, maybe more similar to the treaty than to the Nagoya, and it should be money should be deposited into a global fund. How this will work was not decided on, but is part of an ongoing negotiation. So Amber in her talk will probably go into exactly what is going on in these negotiations. I will therefore skip this one, and then I will talk a bit about another project, or a project that we have been running at our university, which is called the Life Plan Project, which just to sort of talk a bit about how complex this can be. So this is a fairly large project. So it's 130 million kroner, which is 13 million euros approximately, and it's led by Thomas Rothlin at our university. And in Life Plan, Thomas aims to have collection points from all over the world, so almost every country, and he aims to collect samples from insects, fungi, and microbes, sound and images. So basically give a global map over biodiversity as a whole on the globe. in a very sort of fine covering a net. So when Thomas got this money, he also then came up with the idea I actually have to negotiate pick and mats for these, because he didn't have them at the place. So then what happened was that we at our university actually employed Sebastian, who got the task of negotiating all these hundreds of pick and mats that needs to be in place to carry this project on. So Sebastian will continue with this, talking how on earth did you manage to get all these pick and mats in place during this project period.
Yes, thank you, Jens. And maybe you should add that this project has been going on since 2020. So we are like 4 years into this project, and it will end in 2026. So most of the work is actually done, and a lot of lessons has been learned. And I'm going to share some of the insights from working with this pick and mats and ABS contracts with you. We'll now stop your screen here, Jens. And
maybe I will, if there is any questions, I can take that and then I will hand over to you.
Okay.
So far I don't. There's one. It just came now. Are there also mechanisms in place to retrospectively provide monetary or otherwise benefits for already used DSI or any other resources from different countries?
I'm not so when it comes to my spontaneous question answer to that would be no. The thing is that anything that the Nagoya Protocol it only regulates what you accessed after 2014. So from that time and onwards, you have to have these agreements in place. Anything you accessed before that is not regulated, so there is no places. But on there is no sort of mechanism to pay in hindsight. On the other hand, you have to show that you have access to the genetic resources at that time also. So it's not necessarily. I mean, it might be a bit on case-by-case basis, actually, in that respect. But I don't think there is any general regulations in place for that, doing that, no.
Okay, thanks.
Okay, now.
There's actually one more. Just realized as well. I can read it aloud. So someone is asking, can you talk about when in taxonomic research ABS is relevant? Many Nagoya FAQs say things like the identification and classification of organisms is not considered to be a form of utilization under regulation number 511/2014, as long as the research does not investigate the genetic or biochemical composition of the organism. But to which extent does that go?
Maybe can I answer that question, yes, or?
You can do it.
Because I know there is a guidance document about this EU regulation that says that taxonomic work is not covered by the Nagoya Protocol, in the sense that you have to comply with it on EU level, and I think that's correct. But at the same time, just because the guidance document for the EU legislation does not apply to countries outside EU, so you still have to kind of be sure that the things that the work that you're planning to do in countries outside EU doesn't see it any other way. But working with like taxonomy, you may not need to, for example, use the declare. You don't need to declare it within EU if your research is outside the scope of Nagoya Protocol according to this guidance document.
Thank you.
Okay,
Thank
I
you.
think there's no more questions for now, so we can go on.
Can you see my screen, someone?
Yes,
And
yes.
the
Yep,
rest of
it's
you?
all there.
Yeah.
Let's see if it works to. Do you see life plan map here?
Yep.
Yes.
It's kind of yeah. There's an oversight on like in which countries we were like collecting, and we're still collecting samples to be honest. So it was like a major work to find out what's like the rules in every specific country. But it started in 2020. We've been doing this for 4 years, and most of the countries are up and running. We are collecting some samples all over the world, and yeah, we are still have a few countries left to go, but yeah, some are harder than others. I will talk about the ABS contracts today, and as Jens mentioned during his presentation about this, if there is something that you very early on see when you get into this framework of Nagoya Protocol is pick and mat. And when I started with this, I never seen that before. That was something new for me, and I kind of thought like, okay, this is something that I need to get from each and every country. You may also see things like MTA, memorandum of understanding, letter of intent, etc. That is like contracts and words that I'm familiar with from before, but pick and mat is quite unique to the Nagoya Protocol. This pick is a prior informed consent and is most of the time an administrative permit granted by an authority of the provider country that you need to have before accessing the resources that you want to access. But one thing that I realized working with this is far from all countries actually have a pick, and there was a lot of misunderstanding from my side in the beginning because I always kind of approach countries and asking how do I get the pick, and they many times didn't know what I was talking about. So like, yes, there is something called pick, but don't take it for granted that all countries actually use a document that is named pick. They may use something else that they may not have it at all. But that is something that will kind of save you a lot of trouble if you enter into like your country with more open-minded that maybe I did at the beginning. It's also possible that the pick and mat is jointly issued in one document. It could be called, for example, material transfer agreement, where you have the pick information and the MIT as mutual agreed terms in the same documents, and that could also be a bit confusing at the beginning, since kind of I expected it to be one pick and then comes the mat, but that's not always the case. I will actually show you a real pick and how it can look like, but I think I do that in the end of my presentation. Then kind of like I can go through how it can look and what is typical for these documents. Yeah, mutual agreed terms. Almost all the time that is incorporated in a material transfer agreement, since all the time you transfer material from one country to another. So that kind of contract is usually called MTA. Then you add this mutual agreed terms into that contract. And typical things that you regulate in such a contract is like you identify which genetic resources that you're actually going to have access, what you're going to do. I mean, that is like what research are you going to do, terms of benefit sharing, terms on third-party use, and terms of changes of intent. You almost always see this clauses in contracts like this, and I will show you how it can look like after this presentation. And it's like, of course, it would be much easier if there was like a standardized contract that you could use for all countries in all situations. But unfortunately, there is really no one size fits all ABS contract. You kind of have to adapt it at least a bit to each and every situation. But however, it will save you a lot of time, and maybe like for your university or the organization that you're working within, if you could draft some kind of standard contracts that you could use, some model clauses that you can kind of copy and at least take inspiration from when you're negotiating these contracts, because writing everything from scratch takes a lot of time, and it's way easier to have some framework to start from. I should also emphasize that it's said in Nagoya Protocol Article 19 that each party, like they are really encouraging users to build this model contract clauses for mutually agreed terms. But so far, unluckily, I haven't seen much of them. There are some out on the web, but I hope there will be more in the future, because it will be way easier to kind of set up these contracts. Yeah, let's see if I can open up some contracts here, and so you can see how it can look. Let's see. Simon, do you see this now?
Yeah, perfect. It's there.
Can you see when I'm like scrolling too?
Yeah.
Yeah. This is a kind of standard agreement that we use, or we still use, I should say, in LifePlan. And it's mostly it looks like a normal agreement. It's not that special, to be honest. It's like begin with the parties who's going to sign it. Sometimes it's some kind of government authority they're going to sign it. Another time it could be the partner that you're working with, and the authority is just kind of confirms that you have a kind of benefit sharing agreement with a local partner, and that's enough to give you the permit. So you're not always signing agreements with the government. That could also be with like another university, for example. Depends on who you are collaborating with. This is, you write some background, what you're going to do, about a bit about your project. You can take it from the project description that you probably already have. Maybe some small, like shorter description on your organization. And then in chapter 2, you normally kind of write what kind of a material you're going to access. At least we did that. The thing in LifePlan is that we are collecting like thousands of different materials, so we couldn't really identify what material that we were after, and that was kind of a challenge for us. So we made it like an attachment that we are allowed to use all the materials that we collect in a certain way for a certain project, for our certain project, and that's how we did it. But I'm involved in other projects where the researchers, they really know what materials they're looking for and what they want to study, and then you normally put it directly in the contract that this is the resource that is covered by the agreement. And this is mostly like equipment on how to get the materials. It's specific for LifePlan. But then it comes more here on the chapter 5, benefit sharing and scientific collaboration, capacity building. This is typical things that you put in, and it's, in my experience, what MAT, mutual agreed terms, really aims at. What's the benefit sharing in this in the collaboration? So this is pretty unique for this kind of contracts. In our case, we had not only one benefit sharing, we had different kind of, you see there like involvement of local researchers, acknowledgement in the publication, education and training, and so on, provision for equipment, since they could kind of keep the sampling equipment of the projects done. Kind of as a general rule, non-commercial projects offers non-commercial benefits, and commercial projects offer maybe more like money, royalties, and that kind of things. That's as a general term, it's widely accepted in my experience. And then the rest of the contract is a small clause about the intellectual property, confidentiality, liability permission. This is standard contract things, has nothing really to do with Nagoya Protocol. Settlement of disputes, you used to have that in all contracts. And duration, force majeure, protection. So that's more, and then signing. That's how it can look. And this is a contract that we've been using for many countries, and it works fine. And we get the permit, and we get the work done. So I hope this could inspire you a bit on how a contract can look like. Last, I would also like to share how a PIC could look like. It was actually a document that I worked with today. Just a moment. Somebody see this?
Yep.
Yes.
Now there's some like signature there.
Yeah.
This is actually with your home country, Douglas, Kenya. We signed it today, and this is with Kenya Forest Service. We signed a PIC. It's even called prior informed consent, and this is how it looks. Here's the stakeholders, and we are collaborating with a university in Kenya, Egerton University, and we're applying for this together. The Kenya Forest Service hasn't signed yet since we signed today, but as you can see, this looks pretty similar to the contract that I showed you before this. Our standard contract is with a pretty long background, what's going to happen, and the agreement in Chapter 3, partnerships, and then here comes the benefit sharing. Again, it's in Chapter 5. It's typical for these agreements, and a bit like compliance with the law, because there is I have to say, Douglas, Kenya is pretty challenging to work with. There is a lot of legislation, but it's possible if you don't give up. And the signature. So yeah, that's how it looks. I can also, like, as a recommendation, always try to work with a partner in the country that you're going to access the resource from. That is way easier than to try to do it on your own. I think that's it. I hope I didn't take too much time, and there is some time left for questions, perhaps. I don't know.
Thank you very much, Sebastian. Do we have any questions for Sebastian at this?
There's one question.
Yeah.
If Jens, you could maybe provide a brief introduction to Sebastian Bromander.
Okay, so in hindsight, yes. So Sebastian is a lawyer at the Swedish University for Agricultural Sciences based in Uppsala, and Sebastian is a lawyer by training and has been working on these issues around access and benefit sharing to help researchers at our home university, and also been sort of a bit active in helping other universities in Sweden, because at all universities we have to do this job, and with the life plan we got sort of a head start in signing this, in setting up these
I think it's really learning by doing when it comes to Nagoya Protocol. It's absolutely not a perfect system. It's only been around for around 10 years. The countries are doing their own way all over. It's like I haven't seen that no countries do exactly the same way. I hope this will, over time, become easier and more transparent. But so far, it's messy. I got to tell you, it's hard. But you learn by doing, and that's why I'm here sharing today.
Okay, and there's two more questions. Have you had any experience generating PIC and MAT with indigenous groups?
No. Not me personally, no.
And how long does it Sorry, Jens.
But usually, Sebastian, isn't it the pick and mats are negotiated with the competent authorities in each countries?
Yeah, maybe pick, but the mutually agreed terms, like the benefit sharing, especially when it comes to indigenous people, I would say I guess they have an interest to get some kind of benefit for share there, for example, traditional knowledge or whatever it is. But I haven't had any like direct negotiation with such a people, not yet at least.
And how long does it take to conclude pick and mat?
That's a very good question, and unfortunately, there is no straight answer for that. It can take a couple of weeks, maybe a month or two in best case, but it can take you way longer. It also depends on how active you are yourself. I mean, as you saw, we got our permits in most of the country, but we are still, even we've been going on for 4 years, there are some countries that we still haven't had our permit, and we may never get. So it's all from a few weeks up to years.
Okay.
But one thing that you can do is that prepare your own thing, prepare your project description, so you can come up with a kind of presentation. We are going to do this. If you have a local collaboration partner, that will for sure save you a lot of time. It's a lot about trust, and that's way easier to build up with a local partner in the country where the resource is.
Okay, thank you. I see that there is one more, but maybe we can go to the next speaker and save that third question for Sebastian for later, so for sake of time.
Yeah.
Okay, so our next speaker on the list is Amber Schultz, who is the Deputy Director of the Leibniz Institute and also one of the leading persons of the DSI scientific network. That is, and Amber has been thinking a lot about DSI and how to regulate this in relation to an possible access and benefit sharing mechanism. I'm very happy that you are here and can give a talk about this and also share some insights on the ongoing negotiations at the CBD level. Thank you very much, Amber.
Yeah, thanks, Jens. Thanks, Carl Joseph, and thanks to the organizers for this opportunity. I'd like to kind of give you a little bit of a fast fly through of what's been happening in the CBD digital sequence information discussions over the past years and some of the arguments the scientific community was involved in developing. I thought it might be helpful to kind of think about, because this is the Science Policy Forum this week, to think about where and how science intersects with policy. And in fact, this happens across many multilateral environmental agreements. In fact, if you look closely at the titles of all these, I can't actually find one where a scientific perspective or the dialogue between science and policy wouldn't be necessary. And then if you zoom in here, this is sort of our space around the 1992 Rio Earth Summit, where the Framework Convention on Climate Change, the UNFCCC, which most people know about, as well as the somewhat lesser known Convention on Biological Diversity, and its third sister treaty, the Convention to Combat Desertification, were opened for signature. So we're going to zoom in on the CBD, as you've already been mentioned in previous talks, and in particular upon the issue that the CBD had tried to address really in its first sort of 3 decades, namely the focus on access to biological samples, and the term of art in these UN documents is genetic resources. And as Jens already pointed out, the objectives of the CBD are quite simple in some ways, and powerful. The conservation of biological diversity, the sustainable use of its components. But the third objective is probably the least intuitive, or at least it was for me when I began working in this field: the fair and equitable sharing of the benefits from the use of genetic resources. And ultimately, this is part of the grand compromise, the grand bargain, and should create sort of a motivation and an intrinsic sort of valorization or monetization of biodiversity to be able to serve back to the first two. It also is the expression of national sovereignty over biodiversity. Just as for oil, gas, and minerals, the law begins to say you can't just go into a country's border and take and use its biodiversity without asking permission. So indeed, that biodiversity is not under the common heritage of mankind, but belongs, in fact, to that sovereign nation. Why and how did a little collection here in northern Germany get involved in this space in the first place? It's because we do a lot of exchanging of genetic resources. So we send and receive microbial samples or new species of bacteria, archaea, and fungi to 86 countries around the world. We have about 10,000 customers, about 2/3 international and about 2/3 academic and about 1/3 non-commercial, industrial, sorry, that should say commercial. So it was being aware of that responsibility under an international law that we originally got involved in the CBD, and then it's Nagoya Protocol. And then I happened to be at COP14 in Egypt in 2018 and stumbled across this issue on DSI, and the rest is history. But let me first take you through the principle of ABS. So access and benefit sharing is actually quite simple in principle, in theory. It means asking for permission before taking materials. It means giving something back, joint research collaborations, co-authorships, training, in-kind support. And ultimately, all of that package should contribute to conservation and sustainable use of biodiversity. However, as you just heard from the past two colleagues, this is quite challenging. And so our scientific director, Jörg Ofman, and I came up with a sort of flow chart to explain how that process works in principle. And the real challenge comes in here at this middle point. And that is namely that as much as there is an attempt to try to do international standardization, every country can decide how their national access and benefit sharing laws work. So while there is an ABS clearinghouse in which all that information should, in theory, be captured, and there should be an email address, and you should be able to reach out to that national authority, because every single country can do it their own way, and systems are in different states of development. For the user on the ground, think about the map that Douglas showed, that Jens showed. It can be very, very tricky to keep all of those different national systems in your head and working in the process of conducting research and development. In fact, these problems challenges were so great that the German Ministry of Environment was actually able to fund a project, a so-called help desk for academic researchers here in Germany, which we called the Nagoya Protocol Hub Project, to help to facilitate some of the misunderstandings and the challenges that users face. And that help desk remains open. It's a project that I think is really an important contribution to doing our part for access and benefit sharing under the Nagoya Protocol. However, given the headaches that you can imagine can come out of the bilateral Nagoya Protocol-like system, of course, there might be parts of you as a researcher that might say, well, what if we just source using public data from open data instead of skipping the hassle of the Nagoya Protocol? Meaning, is it possible actually now with technology to be able to use open genetic sequence data from open databases and not have to go through this whole legal process and do feed sampling? And this sort of theoretical question is the trigger, is the thinking around the DSI debate. So why is there a call for benefit sharing from digital sequence information? This came up at COP13 in Cancun in 2016. And ultimately, if you look at the chart on the right from GenBank, from one of the large open access databases, you can see that when the Nagoya Protocol was adopted in 2010, yes, there were big open sequence databases, but their exponential growth over time really caught policymakers' attention. Secondly, of course, the biotechnology tools have changed significantly since 2010. One of the biggest, of course, would be CRISPR-Cas9, the so-called genetic scissors that make genome editing very simple. But beyond CRISPR-Cas9, if you look at the table on the left, wherever you see 3 stars in a column, those are different kinds of tools that scientists need to be able to do in silico or computer sort of based and synthetic biology edits to model organisms, for example, or to old material in combination with data out of the public databases to be able to reduce their reliance on physical samples. Now, I will say here as a side note, I do not think that there is any field that can completely avoid physical samples. But the point and the tease for these slides is to trigger your thinking that there is a change fundamentally in the life sciences. And of course, artificial intelligence has made this even more prominent in the last year or two. So ultimately, I think the discussion that was being had a lot through the pandemic and in the years before is that the Nagoya Protocol was simply not future-proof. So you can see that exponential growth doesn't look as dramatic here with a log scale on the left. And this data shows us other kinds of biological data that are managed by biological databases like EBI. And I wanted to highlight, for example, that black line on the bottom right, which might seem not to be doing much. That would include things like protein structures. And here, just to give you a sense of further changes beyond just synthetic biology, CRISPR-Cas9, and genetic sequence data, I think that ultimately we can't quite see what's around the corner, and being able to design policy mechanisms that can account for this is very difficult. So here's an example of an artificial intelligence-based algorithm called DeepMind that enabled those few 100,000 protein structures that were that black line in the previous graph to be able to go up to the next log scale on that, meaning that you had nearly 200,000 sequences, and now you have over 200 million protein structures predicted. So those huge jumps and bounds mean for me, A, that the future of the life sciences will be based upon the use of the global data set, the collective aggregate. And it also means for me that if policymakers want to design a system that's effective, they need to think about what the future of the life sciences might hold. Another important aspect of DSI that was brought up is the issue of interoperability and interconnectedness. So here's a graph of just 19 sequence databases and how they exchange data across themselves. So this issue that is, I think, different than under the Nagoya Protocol for physical samples is that there are hundreds of millions of sequences, thousands of databases, hundreds of data types, and that is extremely interconnected, interoperable, and needs to be done in order to maximize the scientific output that Douglas so elegantly described in his first talk. And the reason for that is that biologists can be active or conducting research in any one of these words on this knowledge graph here. So that flow of data across those databases is really to be able to enable biologists to zoom in, for example, on gene products, on morphology, on chemical structures, on taxonomic identification, on the cultivation of the growth of organisms, on motility, et cetera. And so depending on the scientific question that you want to slice, dice, and reorganize the data in different ways. So fundamentally, the argument that has been made is that while there's clearly a need for benefit sharing from the revolutions that are happening in the life sciences, this system, this infrastructure that delivers information is incredibly important to preserve, to maximize use across the future and across the different disciplines. So really where we are right now from 2022 to beyond, let's say 2050, is that benefit sharing from DSI is needed, but ultimately our advice from the scientific community to the policymakers was the mechanism cannot be Nagoya Protocol-like. So imagine those pages and pages of that MAT agreement that we just looked at being done for hundreds of millions of sequences. It's simply not practical. Another important observation, this is done based on some research done in collaboration with the Leibniz Institute in Gottesgaben, the plant collection there, as well as with colleagues at EBI and at One World Analytics, is noticing that scientists use local DSI, local data, more than they use foreign DSI, and the importance, again, as we heard in Douglas's talk, about having access to local data to solve local or regional problems. So for example, if you look at, let's look at the African group on the left-hand side. So on the x-axis, you have the different UN regional groups. And this is a graph just showing the country of origin for the DSI. This is not where that was sequenced. This is actually where the sampling took place. So imagine a cassava plant virus is certainly in one of that in that graph. That's the bottom. Where did it come from? And then the color is the place where the user, one of the authors in a publication that cited DSI, came from. So you can see that in every single bar here, including the African one on the far left, the biggest color matches the country of origin. So for example, the biggest group of users of African group sourced DSI are African users. And that trend remains for Asia Pacific, for Eastern Europe, for GRULAC, Latin America, and for Western Europe and others. Meaning that while we are very, very interdependent upon each other to be able to solve scientific problems, we also know as scientists that we have to have access to our regional or our local DSI, maybe even more so than that of others. And ultimately, this was one of the motivations for founding the DSI Scientific Network. So this was a network that was founded in 2020. It now has more than 80 members from 28 countries. We conduct online webinars, including ones like these. I should note that both Douglas and Jens are members of the network. We try to attend official CBD meetings, develop briefings and positions papers, and coordinated outreach together. We developed in 2022 a position or a peer-reviewed publication on the need for a multilateral rather than a bilateral Nagoya Protocol-like mechanism, a multilateral benefit sharing mechanism to support science and biodiversity conservation. You can see the authors and the abstract here, and I'm happy to put the link in the chat. That paper really brought forth 5 principles in what we were hoping could be a win-win DSI benefit sharing solution. We called for open access, and in particular, that acronym ABS, access and benefit sharing, actually be decoupled, that the point of access to data, to DSI, not be the trigger point for benefit sharing, that access could be open and that benefit sharing could happen downstream. We also called for simplicity. As you've seen, a lot of the bilateral ABS world is rather complex, and because the data in the space of DSI is so complicated, so big, and growing so fast, simply put, the legal framework cannot be complicated in order for the system to work. We also called for harmonization, meaning that other international treaties, as have already been mentioned, the International Treaty for Plant Genetic Resources, as well as several others, such as the High Seas Treaty and agreements under the World Health Organization that are under development, should also look to the CBD and try to coordinate amongst each other in order to create a system that makes sense for users. Ultimately, all of these efforts should lead toward increased conservation, sustainable use, and green growth, think bioeconomy, and the achievement of the goals of the Global Biodiversity Framework. Finally, we did note that the role of databases very much could be to provide indicators or monitoring kinds of information from the global DSI data set that they hold, and that they are willing and able to do that. However, the databases are not in a legal nor an appropriate position to be database police. And that is to say, to be able to enforce national or Nagoya-like rules across large global databases. To make a long story short, the DSI Scientific Network took the multilateral open access message to COP15. And to make 2.5 long, grueling weeks short, the gavel fell at around 4:15 a.m. As you can see here, people are happy but tired. And ultimately, what they negotiated and agreed upon was the Global Strategic Plan for the Convention on Biological Diversity, known now as the KMGBF, the Kunming Global Biodiversity Framework. It was, at the high-level segment, developed as a take-it-or-leave-it package of 5 items. And here you can see the political interplay between digital sequence information, the 5th item, and the negotiation of large important biodiversity or environmental agreements. So the GBF itself, its goals and its targets, the monitoring framework, which are the indicators, a capacity building plan, and resource mobilization, meaning the financial resources and a global fund. DSI in some ways is a balancing act, just like the third objective of the CBD is. It is the linchpin, or it is part of the giving and taking in political negotiations that enables that entire package to move forward. So what was in it, Jens has alluded to this, the decision basically on digital sequence information is a list of principles that define the parameters or the criteria for how the DSI benefit sharing mechanism will work. There is a clear focus in those principles on open access, as well as improved data governance and equity. The commissioning of 3 studies, of which 1 has been delivered and 2 are ongoing, and perhaps most important for us and for the current negotiation phase is a decision to establish a multilateral benefit sharing mechanism for digital sequence information. However, as much as that is very good news for the scientific community, in my personal opinion, the COP15 decision is still, I think, everything and nothing. So paragraph 16 is that last bullet point. It decides to establish a multilateral mechanism for benefit sharing, including a global fund. But that's all we know about that mechanism. And paragraph 17 is where we are now. That is a fair, transparent, inclusive, participatory, and time-bound process to develop and operationalize paragraph 16, that multilateral mechanism. And there will be, there are already, Douglas and I are both involved in the informal advisory group on DSI, which meets once a month. It meets on Tuesday morning at 3 a.m. We get to do our time equity burden here coming up pretty soon. And is continuing to work on a lot of the tough, outstanding issues that remain before parties. I also wanted to have my last slide on what lies ahead. And although this particular science policy forum, which is in the context of the CBD, has focused very much on the CBD, I wanted to remind all of us that there are other United Nations fora that are on a DSI collision course with each other. So you can see the High Seas Treaty, BBNJ, they're at the top, the Plant Treaty, old, out of scope. There was a question in the chat on older material that was deposited through the WIPO, the World Intellectual Patent Organization Standard 26 procedure. They're listed as patents, as well as a number of viruses that might cause public health emergencies of international concerns, or FICs, that are being currently the definition of pathogen being considered under the WHO Pandemic Preparedness Agreement. And while you look at the right side, those are the legal world, on the left side, you see how mixed together that biological stuff is, meaning that if you're a scientist working on fungi, you need to pay attention to all of these organisms. If you're working on viruses, again, you have to pay attention to all of these. And ultimately, these databases are put together in order to enable the comparison with everything to be able to improve the knowledge that can come out of the system. And so I really hope that once the CBD gets its multilateral mechanism set up and moved ahead, that the other UN fora will work their hardest to create a system that can work across the global DSI data set. Yeah, and a final short little plug that if you want to be part of the ongoing DSI policy outreach, we are open to new members within the network. Please just send us an email and we'll get you on board and look forward to broader international participation. And with that, Carl Joseph, I am through and I'm very happy to take any questions or to engage in the general discussion.
Thank you. Thank you very much, Amber. So do we have any questions now? We have one previously for Sebastian, but I would like to start to see if there's questions specifically for Amber.
Carl Joseph, maybe you can bridge the gap until everybody's done typing?
Yeah, thank you. It's a very general question, in fact. So I think these principles designed by the DSI scientific networks are great. And if they are realized and set into practice, it's fantastic. Do you need more support? So for instance, do you need support by IUBS or other organizations? That's my question. So you are strong by yourself, or you need additional, or you need additional power somehow, and that could be discussed. Yeah.
Yeah, Carl Joseph, what a great question. Thank you. I wish everybody asked me that question. I mean, we do. So this has been a real transition. So the DSI Scientific Network has been funded on a shoestring budget from my research grants here at the Institute, as well as one or two others. For example, the CGIAR puts forth a small sum over the last 2 years. But we've been fighting really hard to stay alive. I will be perfectly honest with you. So we have invested, like I said, research grants and some smaller foundation grants to enable travel scholarships for colleagues from low and middle income countries to attend the international meetings. We also have running secretariat fees to be able to maintain, you know, that mass of people to be able to do monthly calls and working groups and position papers. And it would be wonderful. I mean, I did learn from Jens in November that IUBS does sometimes have these sort of science policy supportive mechanisms, and I would love to reach out to you. And it's not only also about finances. I mean, the logistics are important, and there's some famous expression like money is not everything, but without money, everything's nothing, or something like, right? You do need to have your basics kind of covered, no matter what the perspective is. But the other important, I think, aspect is that in anything with involving the United Nations, balanced and global voices are really important. So, you know, if the white girl from Northern Germany says, I need open access, it's not as powerful if many different men and women and they from many different countries say we need open access because we're dependent upon each other and we want to take advantage of the power of global science. So it is also a call, because the IUBS is an international network of many different disciplines across the biological science. It is a deep, heartfelt plea to have more and diverse scientific engagement in these policy processes. And I think we have learned as a community that if scientists aren't paying attention and involved at the right moments, that well-intentioned policy like the Nagoya Protocol can go a bit awry. Like Sebastian said, I expected that a MAT would be a standardized document. Yeah, I think everybody kind of thought that. But if you were paying attention to the language, there's nothing in the Nagoya Protocol that makes it be that. And that's why every single word in negotiated text matters. And as much as it is emotionally exhausting to be involved in text-based negotiation, and there's a lot of gray hairs and wrinkles that I never would have had if it weren't for DSI. If you wind up 10 years from now being mad that DSI rules are like they are, then it's your fault because you didn't get involved now. So thank you for that question. So the answer is yes, we need money, and yes, we need diverse voices.
Just before the question is asked, that is now in the Q&A session, we are not Bill Gates.
Yeah, no, but if the Gates Foundation is listening, or if any other large philanthropic organization is listening, either now or later, IUBS can give me seed funding, and then, or the network, not me personally. My salary changes not at all. No matter what I do, I get the same public servant salary. Yeah, anything helps.
Thank you very much. So there is a question in the chat regarding that says, how international is the current DSI scientific network? So how diverse are we?
Yeah, no, that's a great question. So I had it very quickly on the slide, I did not update that slide. I think that was updated in January. So right now we have, I think, 26 or 27 different countries that might be closer to 30 now. We have members across Latin America and Africa with, I would say, like 60 to 70% coverage. Of course, like in South Africa, we have, I don't know, 5, 6, 7 members. And then in some Western and Eastern African countries, for example, maybe only 1 or 2 or 3 members. Our weakest coverage is probably in Southeast Asia and the Pacific. That's kind of also reflective of the amount of participation also in the negotiation space, I think it's fair to say. So for example, we have members in Malaysia, in the Philippines, in Australia, Japan, South Korea, et cetera. But we definitely don't have the same level of coverage as we do, for example, in Western Europe or Africa or Latin America. Those are probably our 3 most active continents, which is also a bit of a problem because then we schedule our calls that are more friendly to those time zones, making it harder for those members to participate. But in general, I'm pretty proud of the 2.5 dozen countries that are represented in the network.
Okay, thank you very much.
Oh, I can put it in the link in the link to our members page in the network for whoever asked that question.
Mm-hmm. So also just because we have one old question from Mark Hoffman to Sebastian, actually. Can you read that, Sebastian, yourself?
Yeah, I can. And I sent like a text reply to that same question, like the definition, what is a PIC? Because there is a definition, and I had it in my PowerPoint, but that's not always the case in real life, where it shows out that not all countries use PIC at all. But at least in my experience, when I've been handling PIC, it's always been with an authority in the country, and it's always been before access the resources. When it comes to MAT, that is mutual agreed terms. I kind of never seen a document that is actually named MAT. It's always incorporated either in a PIC or in an MTA or in some other agreement. But that is, it comes most of the time comes down to the benefit sharing terms. That is mostly what it means by experience. I think there was some other question to if it's possible to work without a local partner and trying on your own. I mean, I would say it's possible. I mean, I don't see any regulation out there that says it's not possible, but it will probably be harder, especially with some countries. It's a lot about trust building, and it's hard to come to a country, contact the competent authority and ask, hey, I would like to collect this and this and this to do this and this and that. I mean, it's maybe possible, but it will for sure be harder. But at least there is nothing that says it's illegal to do that, or yeah. And then when it comes to benefit sharing, yeah, I think you could make a deal with an authority to share like benefits with the country. It could be like the result of the study is important for the country, or it's all about negotiation in the end. So I guess you can come up with something there.
Okay, thank you. There's also one question if we share our presentations, or specifically Amber, if you are willing to share. So I have actually one question for Amber also regarding. So in this ongoing discussion about DSI, do they discriminate between sort of monetary? Because I think, I mean, in my understanding, there was the sort of decision to have a global fund or of some sort. But is it also discussion about non-monetary benefit sharing from the use of DSI, and how does that relate to access?
Yeah, I mean, there's no clarity on this yet. So under the CBD and its Nagoya Protocol, there are sort of, as been discussed, 2 types, monetary and non-monetary benefit sharing. And I think there's a growing appreciation for the value and the impact of non-monetary benefit sharing. At the same time, it's a lot squishier or hard to sort of see and understand. So, you know, money speaks, as we just talked about. And certainly that will continue to be one of the big fights. What is a fair and correct place to force maybe especially commercial users to put forth monetary benefits? That's going to take up a lot of oxygen in the room. The non-monetary benefits, unfortunately, probably will take up less oxygen in the room, and so they might be also handled less cleanly, or there would be a negotiation risk in that. And I think there's sort of no harm in it being related to access. I mean, you can't get away from the fact that you need to have access to DSI to generate the benefits, right? So like access is important. The question is whether or not the point of access is the right place to limit or check or control for benefits, or whether or not there are further trigger points downstream that are more appropriate. Ergo, your question on non-monetary benefits is how and where could we learn about the non-monetary benefits that are generated right now that are very diffuse and hard to quantify from the use of, in particular, open sequence data. And in my thinking, there's basically two things that you could do. So number one, and this is what we did in the study that I showed, you can not easily, but you can, on let's say an annual basis or every few years, you can monitor and you can use informatic data science tools to ask the question, how many scientists from which institutions in which countries have benefited from open data because they've cited it in scientific publications? And have they worked together across multiple countries? And what are the fields of research that they have contributed? What is the new knowledge categories that they have contributed to? That's pretty easy to do right now using existing bioinformatic tools. And if you took an inventory like that every year or three, and had a global monitoring of the DSI scientific output delivery measured as a proxy through scientific publications, that wouldn't be hard to do. The second thing I think you can do for those things that don't end up in publications, of which there'll be a lot of non-monetary benefits like that, is the possibility to create a global user-based reporting system, maybe attached or in relationship to existing tools like the ABS Clearinghouse, in which case you as a user could say, aha, I had a grant or I had a project like LifePlan, and we used the global data and we did these trainings and these workshops and we contributed to sustainable development in these countries in these ways. And I fill in an online form, I create some links to my project website, and I say, that's what I did. And if everybody kind of does that, then over time, that data gets collected and can easily be aggregated to sort of show change over time. And interestingly, those kinds of systems are used in other indicators and other goals and targets under the Global Biodiversity Framework. So imagine that data could also be transported back to the National Biodiversity Strategic Action Plan reporting, the NBSAPs, and sort of pre-populated into those reports. And then those countries could report on that if they wanted to, or it could be aggregated at the global level. So the long story short, I think where possible, parties should use the existing scientific infrastructure so that there's not more reporting burden. But where there is no good scientific infrastructure to harness information on non-monetary benefit sharing, I think we might still need some additional new tools that could arise out of these DSI discussions. And I would also beg for those tools to be as encompassing across multiple UN instruments as possible, and ideally.
Mm-hmm.
Thank you very much. Thank you.
Yeah, so maybe I take back the moderation, Jens,
yes.
finish, because Amber, so we are very glad that Amber had time today, because initially we wanted to have the webinar tomorrow, and then Amber would not have joined. So we are very happy that you joined us, and now she has to rush to public transportation or something like that. So I'm thankful to you and to Douglas and Jens and Sebastian and Simon for moderating it, and Natalie for organizing it. And I should mention from Natalie, she just wrote me that the presentations are available online on the IUBS web page. So if you want to listen to the entire webinar again, you can find it online. So thank you so much. I would close here just with showing the advertisement. Okay, that's the wrong one for tomorrow's session. And that is, oh, is it right? This one. So tomorrow we are going to have another webinar, Role of Indigenous Peoples in Conservation and Biodiversity, moderated and coordinated by René Borges, also in the Executive Committee of IUBS, and Sunisa Subramanian. So please think about it. So tomorrow, that's earlier, it's 2 hours earlier. So tomorrow, and you find again all information on the web page of IUBS. So I would stop here if there are no other. I look in all these happy faces. There's nothing left to say, or is there anything to say? Natalie, did I forget anything? No. Then thank you all for joining. I think it was a marvelous presentation, lots of knowledge. Thank you. Thank you so much.
Thank you very much.
Thanks for joining.
Thank you all. Bye-bye.